Okay, good morning students. This is Mohammad Rafi Sheikh, Faculty of Biology. I have 20 years experience in biology, IIT. Now, today we are going to discuss about that new pattern of SSC model paper, particularly biology. Already previously we know that there are two papers in science, that is called general science. Paper 1 physical science and paper 2 biological science. Now, due to this COVID-19, the government of Andhra Pradesh, they merged both paper 1 physical science and paper 2 biological science and combined together, and they make a single paper. So that is general science. So now in this mode of examination, how can we know that how many questions comes from the physical science and how many questions from biological sciences? Now, to make clear about that one, now we are going to discuss about that the new pattern of examination paper. So in this examination pattern of paper, there are two parts are there, part A and part B. Part A related to physical science, and whereas part B related to biological sciences. And then part A, they contain 46 marks, whereas biological science, that is called part B, they contain 54 marks. So this is the new scheme or new pattern of the examination. Both physical science and biological science we are going to write in a single paper. So now I am going to explain that one. So how many different types of questions comes from part 1, that is physical science, and how many number of questions that comes under part 2, that is biological sciences. Okay students, now if you come to this general science question paper, it is a combination of physical science and biological sciences, and this according to the new pattern. So the physical science, which is part A, and whereas part B, which is related to the biological sciences. And here they divide in part A and part B 4 sections, and each and every section, for example, now we are discuss about that physical science. So they have 4 different types of sections. In this section 1, first all they comes under that objective type questions. Each and every question they carry 1 mark, and second section, that is very short answer type. In this very short answer type, which contain that is section 2, and they have 2 marks. In this section 2, we write 4 questions or we attempt 4 questions, and the third one short answer type, which under that section 3. So in this section 3, 4 questions we want to attempt in this examination, and each question carry 4 marks, total 16 marks. And then the last section in this physical science, that is essays and diagrammatic questions. In these essays or diagrammatic questions, so they ask 2 questions, one essay question, another from diagrammatic question. In these 2, so each and every one they contain 6 marks, total 16 marks. So 8 marks, total 16 marks. And then that's why by this total completely physical science, which carry 46 marks. And if you comes under this part B, that is biological sciences. In this biological sciences, so it is also same like that physical science paper. Very first one, section 1, which is nothing but objective type questions. 6 questions he asked, each question carry 1 marks, total 6 marks. And the second section, that is called very short answer. In this very short answer type, each and every question they have 2 marks, and total 4 2s are 8 marks for this section 2. And the third one, short answer questions. These short answer questions comes under that section 3. In this one, each questions carry 4 marks, total 4 questions, which is completed for 16 marks. And then finally the section 4, that is essays and diagrammatic questions. So this is the new scheme or new section what they introduced in this new pattern of the examination. Previously we also have that 4 marks questions in section 4, but due to this combination of the both physical and biological sciences, so the marks are same, but physical science 50, biological science 50, but now this time they divide into 46 and here 54 by the biological science. But in the case of this biological science questions, so section 4, here we want to write 3 questions, and each questions they carry 8 marks. So same, the pattern is same, but they increase the marks, they double the marks from every section. So this is the total schematic or scheme of these physical science and biological science new mode or new pattern of the SSC examination what we are going to attempt in 2020 July. So this is about that one. And now if you take this one biological sciences, so actually the weightage of questions Easy questions, they given 60% of weightage, and at the same time average student can easily attempt for this one, they occupy 20%, and whereas the third category, it is called difficulty questions. So I hope that there is no difficult questions in these biological sciences and physical sciences. So that's why, to overcome the problem, we are creating this new video classes. Those who listen, those who follow these video classes, we are giving assured that one, these third category difficult type questions, there is no difficult, all are easier question if you follow these video classes. So in these difficult type questions, they occupy 20 marks, total 100. This is the weightage of the question paper, both either physical science or biological sciences. So now we are going to discuss about that first model paper of the biology. We are preparing some of the model papers, those who are going to write 2020 SSC public examinations. Here my kind request is that, so those who want to attempt, those who want to going to attempt this SSC 2020 examinations, public examinations, to want to get good marks, they always watch YES&YES publications online video classes and practice YES&YES publications model papers. Now I am going to once again explain the patterns of the examination. So physical science part A 46 marks, biological science part B 54 marks. So total 100 marks. Now this time, first time we are going to write combined physical science and biological science as a single subject. Now in these biological science, total 4 sections. In these 4 sections, first section 1 mark questions, we want to write 6 questions, and section number 2 marks questions, 4 we want to write 4 questions here, and we get 8 marks. And then section number 3, in this one same we are also going to write 4 questions, and each question carry 4 marks, 16 marks. And last very important one and very crucial one, so that is section 4. In this one, total 3 questions. In these 3 questions, question number 15, 16, 17, in these 3 questions they have the internal choice, and then question number 17, they only diagrammatic question. We can choose either 17A or 17B. So only choice is present in the section 4. And now I am going to explain this model paper. First one, section 1. In this section 1, total 6 questions, each question carry 1 marks, total 6 marks. First here he given one of the statement. So I am straw color yellowish colored fluid. I am straw yellowish colored fluid, portion after clotting the blood. Who am I? It is very simple. After reading this question, we get understood that one, after clotting the blood means the question related to the blood. So coagulation, clotting the blood or clumping the blood. Here first one, what is the color of the blood? Straw color. How it is? Yellowish. Straw color yellowish or pale yellowish color. When we find this one, after clotting the blood, after clotting the blood, the answer is serum. So plasma, before clotting the blood, the liquid portion which are present in the blood, that is called plasma. After clotting the blood, the liquid portion, pale, straw, yellowish color substance or water like fluid which is present above the blood, that is called serum. And now the second question here, he given 2 statements. In these 2 statements, first statement is Risha said blood purifies in lungs, and another girl, that is she is Rina said blood purifies in kidneys. So read, focus here. Blood purifies in lungs, and whereas blood purifies in kidney. Here there is a controversy. Mostly we studied in so many classes, so many lower classes, mostly the blood purifies takes place in lungs. And here that in our 10th class, we know that in 4th unit excretion, we detailedly discuss about that kidneys, how the formation of the mechanism of urine and how the blood purifies, in each and everything we discussed. Here, by knowing this statement, blood purifies in lungs and blood purifies in kidneys, the correct exact answer is Rina, blood purify in kidneys. So what happened in, sometimes we may discuss about that blood purifies in lungs. Yes, blood purifies. Blood cannot purify here the carbon dioxide, the deoxygenated blood, when they supply hard supply to the lungs. Here in lungs, the exchange of gases takes place, only exchange of gases. So carboxyhemoglobin turns into oxyhemoglobin. Only exchange of gases takes place in the lungs. Means they remove the carbon dioxide and they added oxygen in lungs. That is only the change that occur in the lungs. And whereas in kidneys, you know that all the different types of the renal artery which loaded with nitrogenous waste material, all the different types of waste material which are present in the blood, they carried by the renal artery and enter into that kidney. Here, by the process called glomerulus filtration, all the different types of waste material which are present in the blood, they removed. Simply we said that blood purification takes place in the kidneys. And now we are going to discuss third question. Name the connecting link between. He given the small boxes. These boxes belongs to the light reaction, dark reaction. So this is these are two stages in the photosynthesis. Photosynthesis, they completed within two stages. First one is called light reaction. Second one is called dark reaction. Here he asked that one, what is the connecting link between? So light reaction, dark reaction. So how the dark reaction continues? For this one, so at the end of the light reaction, ATP, NADPH, adenosine triphosphate, nicotinamide adenine dinucleotide phosphohydrate, nicotinamide adenine dinucleotide phosphohydrate, adenosine triphosphate. These are the two different types of energy molecules are produced here. These ATP and NADPH are the end products of light reaction which are helpful to carry the dark reaction. If ATP and NADPH is not formed in the light reaction, the dark reaction cannot complete it. So that's why we said that, so photosynthesis occurs only daytime. Daytime means up to that, even sometimes the light fails, sunset completed, up to the presence of ATP and NADPH is there in the leaf, they continue the photosynthesis process. So that's why here, which is the connecting link between the light reaction and dark reaction means ATP and NADPH. These two called end products of light reaction, ATP and NADPH, and oxygen also end product in the light reaction, but here these two which are helpful to carry the dark reaction. And that question number 4, which is a eco-friendly group? It is very important one, environmental education or environmental science question. So group A, he mentioned that plastic, chlorofluorocarbons, hydrofluorocarbons, DDT, dichloro, trichloroethane, DDT. So here plastic, CFC, chlorofluorocarbons, hydrofluorocarbons, and next DDT. Second one, group 2, he mentioned that paper, cardboards, market waste or garbage waste. These are the different types of groups. He categorized two different groups. In this one, he said that which is a eco-friendly group. By observing this one, eco-friendly means the material which cannot harm to the environment. They can't create any pollution. This type of material is called eco-friendly material. Here these two, by observing these two, group 1 and group B, we find that plastic, CFC, HFC, DDT, they create some hazard for our environment. They create some pollution problems. And group B, paper, cardboards, market waste and garbage dumps, we can recycle it one and they can't create any environmental problem or pollution. So that's why here our correct option is group B, paper, cardboards, market waste. And question number 5, following placards are prepared in your school for conducting a rally by maintaining the social distance. Name the occasion. So it is very important one. So once again, it is what we are facing currently now, that is given in the form of a statement in the name of the placards. So the following placards are prepared in our school to conduct a rally. So by reading these placards, we know that, so what this rally related to which occasion? What is the first one? Stay home, stay safe. Stay home, stay safe. Now in these last 2 months, we are observing this stay home and stay safe in medias and each and everywhere. And second one, cover your nose and mouth with mask. It is also important one. And third one, maintain the social distance, use the sanitizers. So I think already you get that answer. So what is this one? These are the different types of health precautions. We want to avoid the disease called corona or COVID-19. So all over the world, they suffered with millions of people suffered with the disease. So to overcome this disease, the government and the scientists and the doctors, they suggest the peoples to maintain all this one: stay home, stay safe, cover your mouth and nose with mask, maintain the social distance and sanitize regularly. So these are related to the COVID-19 or corona disease. And last question, define the term of bioaccumulation. Define the term called bioaccumulation. Bioaccumulation. It is very simple question. Accumulation means entry that one to another one. Bioaccumulation means enter the pollutants into organism, environment into organism. This is called bioaccumulation, or enter of the pollutants or enter of the poisonous substances environment into organism. This is called bioaccumulation, and another also there, biomagnification. So entry of these pollutants from one trophic level to another trophic level, or one food cycle to another food cycle. So that is called biomagnification. So bioaccumulation means entry of pollutant into organism, that is called bioaccumulation. The transfer from one trophic level or one food chain to another food chain, that is called biomagnification. So by this we completed section 1. Total 6 questions we want to answer. 6 questions, there is no choice, and each question carry 1 mark. Now we are entering into section 2. In this section 2, the questions start with the question number 7. Each questions carry 2 marks, 8 marks for total this one. First one, he given one of the tabular column, and this tabular he said that one, observe the table, fill in the blanks. Observe the table, fill in the blanks. So this question related to reproduction. First one, asexual mode of reproduction example. He given some of the asexual mode of reproduction methods, and what are the example for this one? First one, bulbs, tubers, roots, leaves. These are all asexual mode of reproduction. Or simply we said that natural vegetative propagation method, simply without any interference of these, without any manipulation of the human beings, they carry their perpetuation or reproduction. So bulbs, example for these bulbs are onions, tubers, potato, and roots, carrot, leaves, bryophyllum. These we want to fill in this example. So what we want to fill this one? First one, bulbs, onions, and next tubers, potato, roots, carrot, and leaves, bryophyllum or ranapala. So the leaf depressions of the ranapala or bryophyllum, small birds are arranged, they are called epiphyllous birds. It is also very important, 1 mark question. Epiphyllous birds, their origin or they produce at the leaf depressions or leaf margins of the ranapala or bryophyllum. So this is about that question number 7. So if you miss any one, they cut half mark. So that's why if you are fill this one, both 4 options is there, each option they carry half of marks, and total 2 marks for that one. Another important one, why Mendel chosen pea plant for his experiment? Gregor Johann Mendel, he is treated as father of genetics, and then he observed that pea plants, he worked as a monk in church, and he conducts several experiments. How he conduct that experiment on the variation, how the characters are transferred from one generation to another generation. In this one, he choose a particular plant that is called pea plant, and next he is a basically mathematician. So why so many hundreds of scientists they works on the variation, but only very first Gregor Johann Mendel got success? Why? Means he is basically mathematician and he select the pea plant. These 2 factors they are important to get success in this case of variation. That's why Gregor Johann Mendel treated as a father of genetics. And here why Mendel chosen pea plant for his experiment material means, here pea plant, it is an annual plant. It is an annual plant, and at the same time bisexual plant. Annual, bisexual. First one annual plant, bisexual plant, and after that one the characters can be expressed freely, means the characters expressed externally. Well defined character, well defined characters, annual plant, bisexual, early predominant, and early hybridization. If you want to introduce any new characters, they can catch very fastly. That is called early to hybridization. Early hybridization, annual plant, bisexual, well defined characters. These are the different types of factors which is chosen to Mendel for his experiment material. So we should write point wise 1, 2, 3. Each and every questions in biology we must write in the form of point wise, not paragraph wise. Never write paragraph wise, always prefer to write in a point wise. Why? Means each and every point they give half mark, either full mark. So that's why if you write full paragraph, it is very difficult to judge your answer. And next one, question number 9. Raghu suffered with hypertension. We heard that BP, high BP, high BP. That is called hypertension. And you know that by observe that one, some of the people, some of the neighbors, some of the brother or elder sister, they ask that one. They want to know that what is BP, and at the same time how it is measured. So now explain what is condition of Raghu. So what if the major question or major idea is what is hypertension. What is hypertension means, if you want to know that hypertension, we want to know one or two important things that is related to that one. So very first one, blood pressure. First this hypertension related to the blood pressure. What is blood pressure means, at which speed the blood flows in blood vessels. At which speed the blood flows in blood vessel, that is called blood pressure, BP. And this BP which is measured by a instrument that is called sphygmomanometer. Sphygmomanometer. So by this sphygmomanometer we measure the blood speed. So that's why this is called what is the BP and how it is measured, and explain the Raghu condition. So if you take that normal blood pressure is 120/80. This is systolic pressure and this is diastolic pressure. So we find two different types of BP. First one is called hypertension, second one is called hypotension. So the blood pressure is this is called 120/80 systolic and diastolic pressure. This is called normal blood normal BP, normal BP blood pressure. So if the blood pressure more than 120/80, this is called hypertension. The blood pressure less than 120/80, which is called hypotension. Hypertension, hypotension. Hyper means more than 120/80 leads to that hypertension. Less than 120/80 leads to the another disease, that is called hypotension. Hypertension, hypotension. More than 120/80 hyper, less than 120/80 hypotension, and 120/80 systolic pressure, diastolic pressure. This is called normal blood pressure. Now by knowing this information, we get understood that one, why Raghu suffered with hypertension. So once again, at which speed the blood flows in blood vessel, that is called blood pressure. And second one, it is measured by an instrument that is called sphygmomanometer. And next, and what is blood pressure means, normal blood pressure is 120/80. More than 120/80 is called hypertension. Less than to the 120/80, which is called hypotension. This is about this question. We should write present one point to another like that. Our next last question in this section 2, that is arrange the following in correct headings. Arrange the following in correct headings. He mentioned, he clubbed different types of body parts. Mostly by reading this we may get understood that one, mouth, lungs, stomach, anus, trachea, alveoli, esophagus, pharynx, duodenum, larynx, small intestine, nasal cavity, large intestine, rectum, bronchi. So by observing this one, what he mentioned the different types of parts, they categorize into two types. Some of the parts they related to the digestive system, some of the part related to the respiratory system. That's why he said that one, arrange the following in correct headings. So after reading this all different types of parts that present in our body, we want to set in a digestive system and respiratory system. We separate these two. And before that one, so here mouth, stomach, anus, esophagus, pharynx, duodenum, small intestine, large intestine, rectum. What I make underline here, these are all comes from digestive system. Stomach, anus, esophagus, pharynx, duodenum, small intestine, large intestine, rectum also. Rectum. So we write all these parts here. Before we are entering here, we want to write in a sequence manner. And the second one, so lungs, trachea, alveoli, larynx, pharynx, nasal cavity, bronchi. they are related to these are the parts which are related to the respiratory system. To know the detailing, our digestive system, they have several important parts. So, first one, our digestive system start with mouth, oral cavity, or it is also called buccal cavity, mouth, oral cavity, pharynx. So, pharynx, it is a common passage. They are present both at a respiratory system and digestive system. It is a common passage for respiratory system and digestive system. So, now we are discussing about that. We are arranging a sequence manner of these are all parts in digestive system: mouth, oral cavity, pharynx, esophagus, stomach, small intestine, large intestine, rectum, anus. These are the parts that related to the digestive system. Once again, mouth, oral cavity or buccal cavity, pharynx, esophagus, stomach, small intestine, large intestine, rectum, anus. So, that is why we want to present here one by one in a sequence in order. And second one, if you comes under the respiratory system, the human respiratory system consists of some of the important parts. They are called nostrils, nasal cavity, internal nares, pharynx, larynx, trachea, bronchi, bronchioles, and finally lungs. Lungs means they contain alveoli. So, here alveoli is called structural and functional unit for lungs. So, these are the different types of parts that present in a respiratory system. So, we can't enter into zigzag manner, but we want to arrange in a simple sequence in order. Once again, so mouth, oral cavity, pharynx, esophagus, stomach, small intestine, large intestine, rectum, anus. And if you are come into the respiratory system, nostrils, nasal cavity, internal nares, pharynx, larynx, trachea, bronchi, bronchioles, alveoli, or it is present in a lungs. So, we want to arrange this one. The part what we miss here, we do not bother. What he arranged, what he given, we may arrange in a sequence order. So, this is question number 10 for 2 marks questions. By this one, question 7, 8, 9, 10, total 4 questions are present in the section 2, and each questions carry 2 marks, and then we may get 8 marks. Each and every one we should write in a point wise. Now, students, we are going to discuss about that section number 3, question number 11. Here each questions carry 4 marks. Sometimes he may ask the diagrammatic questions, sometimes may ask the general question. So, sometimes he may give on the diagram, and based on the diagram he frames some of the questions like this one. So, it is better to choose like this type of questions. They may have very less answers, means we may completed our total question within 4 or 5 minutes. But in this section 3 there is no internal choice. We must write, we must attempt each and every one. And now we are going to read that question. Observe the experiment and answer the following questions. Observe the experiment and answer the following questions. In this one, by observing this one, this diagram or this experiment related to that photosynthesis. Photosynthesis, particularly carbon dioxide is essential for the process of photosynthesis. Carbon dioxide is essential for the process of photosynthesis. In this experiment, he given the experiment, and last finally the leaf what he kept inside these jar also. And now we are going to read that first question. Name the experiment. Above experiment related to this photosynthesis, particularly carbon dioxide is essential for the process of photosynthesis. How can we prove that carbon dioxide is essential for the process of photosynthesis? This is the first answer. So, the experiment related to carbon dioxide is essential for the process of photosynthesis. We are going to prove this by experiment. And second one, why we destarch a plant before commencement of the experiment. So, not only this one, how sunlight is essential for the process of photosynthesis, and before the starch test, we are conducting each and every one, whether photosynthesis occurs or not. Before that one, we keep the plant in dark room. Why shall we keep the plant in dark room means? So, to clear the starch, reserve food, to clear the reserve foods. Here we are going to observe that process called photosynthesis to measure that perfectly, to know that result perfectly. If we are conducting without keeping in dark room, we can't get any proper or maximum or a particular result. To overcome that one, so which experiment we are going to conduct? Particularly light is essential for the process of photosynthesis, carbon dioxide is essential of the photosynthesis. In these two experiments, before commencement of this experiment, we keep the plant into dark room nearly 10 days or 8 to 10 days. What happened in these 8 to 10 days means the material, the starch or the reserve food which are present in the leaf utilized by the plant, and on the day of experiment we surely said that the plant is free from starch. On the day if you conduct, we may get exact, correct, proper result. So that is why we de-starch the plant before commencement of the experiment to get clear exact result. And next one, write any two important precautions for this experiment. So what are the important precautions we choose for this experiment means, so very important first precaution is that to choose a long and narrow leaf. First point, important precaution for this experiment, we choose the long and narrow leaf. The plant should have the long and narrow leaf. Why should we particularly insist on long and narrow leaf means, it is easy to insert through this vertically spilted cork and a glass jar. Otherwise the leaf is very wide, we want to bring that much of big jar. To overcome that one, before commencement of the experiment we choose that long narrow leaf potted plant. Another precautions, write any two important precautions. Very second one, the leaf should not contact with the KOH solution. In this glass jar we find that, so we fill with 5 to 10 ml of KOH solution. The major use of this KOH solution to absorb the carbon dioxide which are present inside the glass jar. To overcome that one, so to overcome that one we use carbon dioxide, they absorb the carbon dioxide, KOH solution. And finally here important one, the leaf should not contact with KOH. What happens if the leaf should contact with KOH? At the time the leaf collect the carbon dioxide which are present in the KOH and they perform the photosynthesis. To overcome that problem we should take important precaution, another important precaution, the leaf should not contact with the KOH solution. These two are important so precautions. And last one, name the scientist who conduct this experiment. Hugo Mole. Hugo Mole, he is a botanist, he conducted this experiment, Hugo Mole. So that is why we are said that Mole's half-leaf experiment. The name of the experiment is called Mole's half-leaf experiment. Only we are using by observing this, half of the leaf turn into blue-black color, left half of the leaf cannot turn into blue-black color. That is why on the name of the scientist Mole's half-leaf experiment, Hugo Mole's. On his name he conduct that, he is a botanist, he conduct this experiment. So first one, name the experiment means carbon dioxide is essential. Second one, why we de-starch means to remove all the starch, stored starch to get exact result. And third one, write any important two precautions means the leaf should not contact the KOH solution. And we select narrow long leaves. And last one, moles conduct this experiment, so that is why this is called Mole's half-leaf experiment. By this we completed 11th question. We should write each and every one point wise, or what he asked A, B, C, D like that, A, B, C, D serially. And next one, if you come this side, question number 12 in section 3, here he given this passage. In this passage, read the following passage and answer the question. He given a long means small passage. By reading, by focusing, by concentrating reading this passage, we may get total answers. So this type of answers are very easy. Questions are present and the answers are also present in this passage. If you are focused on this passage, if you read thoroughly. So now we start that passage. Tarun addicted to alcohol and smoking consumption after a long time. So Tarun addicted to alcohol and smoking consumption. After a long time, he find swellings in hands and feet. He always feel tired and inactive. Doctor said his internal organ was damaged. So doctor suggested a temporary method. This type of method, temporary method to him to clean blood, but Tarun interested in a long-term permanent solution. This is the passage he given very simply, and by reading this one we get that clear idea that. So here what is the passage means, Tarun addicted to the alcohol and smoking consumption. After a long time he finds swells in hands and feet. He always feel tired and inactive. Doctor said his internal organ was damaged. So doctor suggested a temporary method to overcome this problem, but Tarun is not interested in the temporary method or temporary solution. He want to know that he want to take a long-term permanent solution for this disease. Now by knowing this one, we may get that a clarity or clear idea about that the passage related to kidneys. The passage related to kidneys. The symptoms what he mentioned, that is also called uremia disease symptoms. Now we continue the question. First one, what organ mentioned above passage? So which organ mentioned the above passage means they are the kidneys. Kidney. So above passage he mentioned about the kidneys. And second question, name the disease by observing the symptoms: swelling the feet, swelling the limbs, weak and inactive, he can't participate any physical works. These are all the symptoms that related to the kidney disease, that is called uremia. And here uremia also we can define like that, complete irreversible functions of the kidney. Complete irreversible functions of the kidney, it is also called ESRD, also called uremia. So at the time the patient's blood totally, his body parts, particularly hands, feet, they swells and weak, inactive, tired, he can't do any work. These are all the symptoms when you find that the patient suffers with ESRD. Complete irreversible functions of the kidneys is called ESRD, and that stage or the disease is also called uremia. And next one, which method suggested by the doctor? Here what method suggested doctor? So the doctor suggested a temporary method. So he asked the third question, see which method suggested by the doctor. Here which is the temporary method for to overcome the kidneys problem? The process is called, or the method is called dialysis, hemodialysis. So dialysis, by this process we can purify the blood depending on the severity of the patient. If you suffered severely, at the time every 10 days once we can totally collected the blood and sent into dialyzer, and after that one here they remove the waste material and purified blood once again inserted to the body. And this method is called here dialysis. We carry the dialysis with the help of dialyzer. And next question number D. Dialysis. Question number D. What is the permanent solution for the disease? But Tarun is not interested in the temporary solution. He want to know, he want to kind of carry that long term permanent solution. So you know that what is the long term permanent solution? It is kidney So what is the last question? Means what is the permanent solution? Means permanent solution is that only the transplantation of the kidney. We collected the kidney from a cadaver transplantation. Cadaver means brain dead persons or other persons, and then we transplant that, we remove that old kidney which is damaged, which is not function properly. Sometimes it will be left, they can't make any harm to our body, and then we are attached the new kidney to this one. This is the permanent solution for the kidney failure patients. So first answer is kidneys, and second one the disease by observing the symptoms, it is uremia, and the temporary method is called dialysis, and the permanent method is called kidney transplantation. By this one, simply by reading the passage, by analyze, by understood that one, we can get answers in this passage only. So this is very easier questions when we concentrate, otherwise we may get confused here. Okay. Students, now we are discussing about that question number 13 in section 3, means 4 marks questions. In this 4 marks questions, you may ask this question: define the term phytohormones. Write the hormones and their functions in a tabular column. So what is the term called phytohormones, and what are the different types of phytohormones are there, and what are their important functions? We should keep in a tabular column. So it is very easy question, and already many times we practiced phytohormones. And next alkaloids. These two tables are very important one. Each and every model paper we should expect any either this one or another, that is called phytohormones, or another one different types of alkaloids. It is important question. Now what is we want to define the term called phytohormone, and all the different types of phytohormone we should write in a box. Now first one, we want to define the term called phytohormones. So what is hormones? Means the substances which act in minute concentration and control, coordinate the growth process is termed as phytohormone. Very simple once again. So the substances which act in minute concentration and control and coordinate the growth process, these as termed as phytohormone. So these termed as hormones. If they are present in plants, they call phytohormones. If they are present in animals, they call zoohormones. So the hormones which are present in plants, they call phytohormones, and at the same time the hormones which are present in animals, they call Jew hormones. So both hormones are different in plants and animals. Those present in plants they call phyto hormone, those present in animals they call Jew hormones. The substances which act in minute concentration, control and coordinate the growth process is termed as phyto hormone. And after that one, here we have 5 different types of phyto hormones already we completed. So in our syllabus, what are the 5 different types of hormones means? First one, abscisic acid, auxin, cytokinin, ethylene and gibberellin. We want to, he asked that one, draw a tabular column. So these 5 different types of phytohormones we should be interpreting these tabular column. First one, we prepare a table, and after that one we write one by one. First is abscisic acid. So these abscisic acid and their function, it is important function. So many other functions are also there, but I mentioned only the important one. So closing the stomata, abscisic acid which is helpful for closing the stomata. You know that what is the use of closing stomata means at unfavorable conditions. So the plant means particularly leaf, they closing opening by the stomata, they control the process called photosynthesis. And second one, auxins. So auxins, so many auxins, so many IAA, it is also called IAA, indole acetic acid, auxins. And so many artificial auxins also there, IAA, IBA, NAA, indole acetic acid, NAA, naphthalene acetic acid, IBA, indole butyric acid. But here we should mention only auxin, indole acetic acid. The scientific name of the auxin is called indole acetic acid. Here what is the important functions of this one means differentiation in shoots and roots. They make the differentiation at the time of zygote development, the differentiation takes place. Some of the part of the seed they are going to develop as a roots and some of them going to develop into a shoots. So that's why the auxins they are helpful to differentiation the roots and the shoots. And apical dominance, other each and everything which is under control of this auxin, apical dominance, that is also. And next cytokinins. Cyto means cell, kinins means division. So cytokinins which are helpful for division of the cell. So the growth and development for the purpose of this one, always the cells they participate mitotic division. So this divisions, all the divisions are under control of the cytokinins, and it is also helpful for opening the stomata, and at the same time cell division. And fourth one, ethylene. Ethylene is very important one. It is helpful for early ripening of the fruits. So ethylene is helpful for early ripening of the fruits, and large amount of auxin, the high concentration of the auxins that also leads to produce ethylene, and by this ethylene we make it helpful for early ripening of the fruits. And last one, gibberellins. So gibberellins actually it is extracted by Gibberella fusariae virus, it is a fungus. Gibberella fusariae fungus we are extracted by the gibberellins, and plants normally gibberellic acid is present. And these gibberellins are helpful for sprouting the buds, elongation of the stem and breaking the dormancy. So many important functions are under control of this one. And so at olden days, nowadays also they are using the gibberellins for seedless fruits, that is called parthenocarpy. It is a term used for seedless fruits. Parthenogenesis means it is one type of asexual mode of reproduction. So here gibberellins, the major importance of sprouting the buds, elongation of the stem and breaking the dormancy, and they are helpful for seedless fruits also, it is helpful for that one. By this one we can prepare a tabular column and we define the term called phytohormone. The substances which act in minute concentration, control and coordinate the growth process is termed as a hormones. If they are present in plants they call phytohormones. If they are present in animals they call Jew hormones. We find majorly 5 different types of phytohormones. They are abscisic acid, auxin, cytokinin, ethylene and gibberellins, and we mention their functions in the tabular column. By this one we can easily get out of 4 to 4. Definition 1 mark, and then next the explanation about this one. These 5, if you are mentioned that one, we may get out of 3 and totally 4 marks easily. So this is about that question number 13. And now we are entering into question number 14. Here simply he given one of the pie chart, he can't mention any one. He said that one, observe the pie chart, write some important stages and their functions. So it is also one of the important questions. By observing this pie chart, we know that this pie chart related to the cell cycle. So in this cell cycle means the cell cycle is a time, the cell cycle is a period in between the 2 successive cell division. If there is two successive cell division, the gap between the two successive cell division, which is also called cell cycle, or it is also called interphase. Here the cell which is participate in the cell division, what they lose that energy and other body material, they want to regain once again, they are going to ready to participate another cell division. So that's why simply we said that cell cycle, it is also called interphase. Interphase means resting period. The successive, so the gap between the two successive cell division, that is called cell cycle. Here once again they regenerate all the bodily material and other one. So by observing this one, G1 S G2M, this is the right sequence. Sometimes he given this G1 S G2M and arrange it in proper sequence. So G means gap, S means synthesis, gap 2, and last one mitotic cell division. So G1 S G2M, G1S G2M, this is the sequence for the cell cycle. And now first one, what is going on in this one? G1 means gap 1. What happened in this gap 1 means cell size is increased. Already we know that they previously participate in the cell division. Due to that one, they lose some of the mass of their body and some of the energy. To overcome that one, here G1 means cell size is increased. And at the same time here starting the DNA replication, the DNA which are divided, and once again they starting the duplication of the DNA, starting the DNA replication in this G1. And second one, S means synthesize. So DNA synthesis takes place in this synthesis process. So it is also called duplication of the DNA. DNA synthesis started at G1 stage, whereas and total synthesis of the DNA completed in synthesis phase. And the third one, G2, gap 2. So in this gap 2, this is end of the DNA replication. So the replication or the synthesis of the DNA which is started to first G1 stage, totally completed in S stage, and finally the end is occurs in this G2, gap 2 stage. So end of the DNA replication. And the last one, M means it is now ready to another mitotic cell division. M means, M stands here for mitotic cell division. By the regain the energy, by the regain the body parts what they lose in previous mitotic division, they are ready to, they are prepared to ready to participate another mitotic division. So that's why simply we said that cell cycle, it is a place, or it is also called interphase in between the two successive cell division. They regain all the material what they lost by previous cell division, and it is going on to ready to continue for the next mitotic cell division. So this is about that G1S and G2M. By this pie chart we can explain detailedly by this one. Each one carry 1 mark here, by totally 4 marks for this question. Friends, we are going to discuss about that section number 4 and question number 15. In this one, 15, 16, 17, these are the 3 questions that comes under that section 4. Each and every question carry 8 marks, and we choose either here is internal choice is there, we can choose any question which is we know very well. So here the question number 15A, how can you prove water is transported through the xylem? How can you prove that water is transported through the xylem? So before that one, we want to know that so in plants, vascular bundle, they have 2 different types of tissue, xylem and phloem. This xylem, they conduct water, whereas phloem, they conduct nutrients or food material. Here he asked that question, how can you prove that water can transport it through the xylem? So this experiment we can explain with the help of an experiment with that is called Yassin experiment. In this process we follow all the total experimental procedure. Why means it is 8 marks question. So if he asks any 1 or 2 questions, we may complete it, but we can't get any complete marks. So for this to detailed explanation about for this question, that's why he keep in this experiment, and this is for 8 marks question. So that's why we want to explain detailedly in the name of the procedure. First one, any experiment we follow, these are all different types of steps. First one is aim, apparatus, procedure, observation, result, precautions. So these are the stages that present any experiments which are related to our biological sciences. So here according to that question is our aim. I am going to prove that xylem conduct to water or xylem transport to water. I am going to explain with the experiment xylem conduct or xylem transport water. So that is our aim. And second one, apparatus. What are the different types of apparatus which are required to carry this experiment or to maintain this experiment means first one big beaker which contain nearly half liter or 250 ml, that is enough for that one. And second one, we take that stand, this is stand to hold the younger plant, and next one stand, beaker and Yassin plant and Yassin that is comes under the chemicals, and the small balsam plant, the young balsam plant. So by that we can observe the stem is have proper appearance, means younger plant. Simply we said that younger plant, younger balsam plant we take for this experiment. So once again, what are the apparatus we are used here means beaker, stand, balsam plant. And next one, what is the procedure means by taking this selecting of this balsam plant, we take a beaker, and before we are keeping this one with the help of the stand, we add some Yassin. Yassin means Yassin solution means they are due to the presence of bromine, they give red in color, they give red color. So that's why the solution which what we take in this beaker, they are slightly red in color by due to the presence of bromine in this one. After that one, with the help of the stand, we tie up that younger balsam plant by like this, what we are arranged on this diagram like this. And after that one, we keep enter this setup without disturbance for 2 to 3 hours. So that is a procedure. First one apparatus completed. Next to procedure take a healthy younger balsam plant, and then filled with the beaker with water and adding some amount of Yassin solution. Due to that they turn into red color, and then what we are showing in this blackboard, we are arranged the same like that with the help of the stand. And then after that, what is the observation means I observe that after some time, after 2 to 3 hours, the leaf, the stem, what we find here, they are turned into slightly red in color, and at the same time the veins which are present in the leaf, they are also turned into red in color. So that is the observation. And then what is the result means after that one we detach or remove the plant from the beaker, that is Yassin solution, and separated. And after that we observe the transverse section of this plant or the stem, we find that the xylem tissue, they are in red in color. The xylem tissue, it is in red in color. So that is the result what I observed in this experiment. And precautions means we selected not a hard plant, only younger plant which have proper appearance have the stem. So shoot or the stem which is have younger one. So this is very simple experiment. By this one we can prove that how the xylem tissue conduct the water, how the xylems conduct the water, or how the water transported through the xylem tissue. Once again I am reviewing in glance. So first one, so according to this question, transportation through the xylem water means here by this experiment, that is called Yassin experiment. With this experiment I am going to prove this how the xylem conduct water, and we follow this experimental process steps. They are called aim, apparatus, procedure, observation, result, precaution. First aim, I am going to prove that with the help of balsam plant, so xylem tissue transported water, and second one apparatus, balsam plant, beaker, water and stand. And next to procedure here, what is the procedure means before procedure we want to mention one more step here, that is chemicals. We are using chemicals, that is Yassin here, Yassin. And next one procedure means take a young having tender stem balsam plant, and then we keep in a beaker filled with a Yassin solution. With the help of stand we attach this one, and then enter setup we keep in sunlight without not sunlight, we keep in some place without any disturbance 2 to 3 hours. After that one I find that stem is slightly turned into red in color, and the veins of the leaf they also turn into red color. After cutting this one, we find the xylem tissue which is in red in color. By this one the xylem tissue transported water. We are clear about this experiment. It is very simple experiment, but if you follow the steps and at the same time by drawing the neat label diagram, we may get experiment 4 marks, and at the same time this picture or the diagram or the experiment what we are conducted, they also carry 4 marks. So by this one we can easily get out of 8 to 8-1. And now we enter into 15th B. This is 15A question, that is water transported by the xylem. Now 15th B, we can choose either 15A or 15B, which are easier for us. Now here 15B question, now I am going to explain simply what are the evidences of evolution. Explain the homologous and analogous organ. So what are the evidences of evolution? Evolution means, so evolution. So we know that for this one we explain that different types of evidences. Basically in our textbook we know that evidences of evolution are 3 types. They categorized into 3 types. So homologous, analogous organs, fossil evidences, embryological evidences. By giving this simple flow chart, We may get good impression, those who evaluate your paper, by observing this one. Each and every student, they always they write in point wise or paragraph wise answers. That is not good, that is not favor to get good marks. If you mention these simple flow charts, each and everything, the examiner, those who value your paper, he know that one, the student does know something about that particular question. So it is create some good impression on you, it will be helpful to get good marks, means out of to out of. So better to follow this simple flow chart. We know that there are three differences of evolution. First, evidences of evolution. First one is called homologous, analogous. Second one, fossil evidences, and third one, embryological evidences. By this observing this way, evolution means how the organism formed from one to another one, how the unicellular organism to develop a multicellular, simple single cell organism to having that millions and trillions of highly developed organism like human being, how this evolution takes place. For all this one, we need some evidences. Without evidences, no one can agree any theories. That's why here, by observing these three different types of evidences for this evolution, we are going to explain this. But he asked that one, explain the homologous, analogous. Now he can't ask all the different types of organs. He asked only homologous, analogous. So first, this flow chart, they may get at least 2 marks, and now what we are going to explain this homologous, analogous, they are helpful to carry the left 6 marks. And now first one, what are homologous? Homologous and analogous. Homo and analogous. What is homologous and analogous organs? Homologous, analogous means homologous organs means they are the structurally same but functionally different. Structurally same but functionally different. So this type of organs are called homologous organs. And what about that analogous organ means they are structurally different but they carry the same type of function. So this type of organs is called analogous organ. Now we are going to explain this detailedly. First one, homologous organs. So homologous organs, to want to understand this question, first we observe that 4 limbs of man, 4 limbs of cheetah, 4 limbs of whale, and 4 limbs of bat. 4 limbs means upper limbs. So all they are, if you are observe that one, they have the same type of structure. They have the same type of structure, but they carry different types of function. So for example, if you take our hand, which is helpful for grasp or hold this one, and at the same time the 4 limbs of the whale, which are helpful for swimming, and 4 limbs of the bird, which are helpful for flying, and then 4 limbs of these cheetah, which are helpful for running. So these are the different types of functions, but all they appear, they have the same structure. Size is different, size is not matter, but the structure is same. So that's why this type of organs is called homologous organs. This type of homologous organs, they lead to divergent evolution. Divergent evolution. That is about the divergent. And whatever the second one, that is called analogous organs, we keep subheading analogous organs, and then below that one we are going to mention each and every answer in the name of point wise. So analogous organ, it is quite different to the homologous. They are the structurally different but functionally same. This type of organs is called analogous organ. Structurally different but functionally same. This type of organs called analogous organs. In this analogous organs, we observe that some of the example, the wings of the bird and the wings of the bat. The wings of the bird, so if you find these analogous organs in examples of bird and bat, they both are quite different. Bird wings, they covered with feathers, large number of feathers covered by these bird wings, and whereas this one bat wings, they don't have any feathers, they covered with skin, that is called patagium. Patagium means elastic like skin which is covered by in between the fingers. Our fingers are individually present, but in these bat, the skin which is covered by these fingers, that is called patagium here. And now analogous organs means structurally different and functionally same. Here if you observe that what the structurally different but functionally same, birds wings covered with feathers, bat wings covered with skin, patagium, but finally both are helpful for flying. They are helpful for flying, means they carry the same function but their structure is different. So this type of organs is called analogous organ, and they lead to convergent evolution. They lead to convergent evolution. So this is about the total the question, what are the different types of evidences of evolution. and explain detailedly about that homologous analogous. Simply once again in the glance. So we want to mention this flow chart. This flow chart give idea that what are the different types of evidences for evolution. And this one he asked that first one homologous analogous by keeping the heading subheading homologous organ. Homologous organs are they are the structurally same and functionally different. This type of organs is called homologous organ, and we present that following one point wise. For example, if you observe that 4 limbs of a man, 4 limbs of a bird, 4 limbs of a cheetah and 4 limbs of whale, they have the same type of arrangement. All the bones and other they have the same type of structure, but the size is different. Size is different, that is another one. Structurally same. And then if you are observe that function, they have the different in function. We are helpful for our hand to grasp or hold, whereas the cheetah for running and whale is for swimming, bat for is flying. So these are the different types of functions, but structurally same. We write as a point wise this one, and these homologous organ leads to the divergent evolution, and whereas second subheading, that is analogous. Analogous organs is they are structurally different but functionally same. And we give the example, the wings of the bat and wings of the birds, they have the different in structure but they carry the same function, that is called flying. And then the skin, what we said that in between the fingers they call patagium, it is important one. And after that one, these analogous organs they lead to the convergent evolution. Homologous organs they lead to the divergent, analogous organs they lead to the convergent evolution. By this one point wise we should complete the answer. So first we want to mention the flow chart, second one subheading of the homologous, definition and examples, and third one analogous organs, subheading and definition. After that one we should support our statement to how it is called analogous organ, what are the examples we should mention in the point wise. For example, bird and bat wings. So by this giving this type of explanation we may get out of 8 to 8 marks. Flow charts, point wise answers, simply required diagrams are very important for 8 marks question. Why? It means it is a descriptive or essay type questions. Here we need elaborate explanation. So by keeping the time in mind in maintains, we should present all this one by one in the point wise manners, and diagrammatic any related diagrams we must mention here. And at the same time flow charts also very important. They are the key points in our biology. By keeping all this in view, you should complete your answer. Surely you get 8 out of 8 to 8. Now students, we are discussing about that question number 16a. In this question number 16a, the question is how the breathing and swallowing controlled by the epiglottis. A very important question. How the breathing and swallowing. So breathing related to the respiration, swallowing related to the digestive system. So how the epiglottis coordinate both respiratory system and digestive system. So for this one we want to know that what are the different types of parts in respiratory system and what are the different types of parts that present in digestive system, means particularly up to esophagus. That is enough here. And now before we are going to discuss how the respiratory system and the digestive system up to what they take into the up to the common passage, that is called pharynx. Up to the pharynx it is enough. Now here first one we are going to discuss about that breathing. Breathing means it is related to the respiration, nostrils, nasal cavity, internal layers, pharynx, larynx, up to this one. And next one, swallowing means if you take that one, mouth or buccal cavity, pharynx, and next esophagus. So by this one, the division in between the digestive system and respiratory system we want to conclude here in the name of our answer. So first, before we want to know that we have a simple explanation to understand this one. When pharynx connected to the larynx, pharynx it is a common passage for food and air, and larynx it is a passage of the gas. So when pharynx connected to the larynx, here the digestive system, respiratory system, digestive system both are separated, both are divided with the help of a muscular flap. So that's why when pharynx, this part is called pharynx and this part is called larynx. When pharynx connected to the larynx, at the time we find that small muscular flap is present. When pharynx connected to the larynx, that area is called glottis. Above the glottis, a small muscular flap is got there. This muscular flap is called epiglottis. It is a very important structure. This structure they divide the respiratory system and digestive system. And simply we said that one, this epiglottis it's act like a traffic constable. How the traffic constable regulate the traffic from one by one to overcome the problem called traffic jam. Here is also the food and air both are entered into the common passage that is called pharynx. After that one, these epiglottis which act like a constable, trophic constable like that one, they divide that food into esophagus, whereas gas into trachea. So that's why where the pharynx connected to the larynx, a slit like opening is there, that is called glottis. And these glottis, they got by a muscular flap. This muscular flap is called epiglottis, and these epiglottis separate the food and gas. Now we observe these two diagrams. First diagram shows that breathing, respiration, whereas the second diagram represents to the swallowing, means digestion. What happened when we eat the food and what happened when we inhale the gases. So first one, this area which is called nasal cavity. So oral cavity, this is oral cavity or buccal cavity, and this is nasal cavity, and here this is the digestive system part, it is a respiratory system part. Oral cavity and nasal cavity, they separated with the help of a muscular flap or a bony plate. This bony plate is called palate. This bony plate, they separate nasal cavity and buccal cavity. And then here after that nostrils, if you are inhale the gas, they enter into nostrils, nasal cavity, and then internal layers and pharynx. When they enter into the pharynx, here the part which is called here glottis. These glottis, they got by the muscular flap epiglottis. These epiglottis, they stand erect. Means normally they present like this condition whenever we are eating and whenever we are breathing. Normally, mostly, always, why means throughout our day and night we are taking only 1 or 2 hours to eat, and left always we are taking the respiration. So that's why always these epiglottis, they stand erect, muscular flap, they stand erect. So at the time the gas directly enter into trachea. How the means nostrils, nasal cavity, internal layers, pharynx, larynx, trachea, bronchi, bronchioles. So the gases enter into lungs. So at the time the epiglottis stand erect. And second condition, if it comes to this digestive system, in the case of swallowing, so whenever we take that food, after thoroughly masticated by the teeth, the food, the bolus enter into esophagus, the bolus enter into pharynx, and pharynx into esophagus. Actually they want to enter into esophagus. At the time, if you observe that the passage, the epiglottis stand erect, due to that one, that air directly enter into trachea at the time of respiration. At the time of digestive system, at the time of we are swallowing that food, the muscular flap, you find here very clearly, the muscular flap, they fall or they close the passage of the trachea. They close the passage of the trachea and then they allow the passage continuously to this esophagus. Means by this one, the food whatever we are swallowing, that one, mouth, pharynx, buccal cavity, pharynx, the bolus, they directly enter into esophagus. So by this one, by observing this here picture 1 and picture 2, the movement of the epiglottis, here they stand erect, so they allow the gases directly enter into trachea, and whereas this one, they block the passage, the respiratory passage blocked by this one epiglottis. So that's why you find here very clearly they fall on these the muscular flap, means they close the passage. By closing that one, the food is directly enter into esophagus. By this way the food is directly enter into esophagus. So that's why the major important structure, that is called epiglottis. The question, if you read that one, how breathing and swallowing controlled by epiglottis. So this epiglottis play an important role to separate food and gases by act like a trophic constable. They regulate the both food and air. Always by standing erect, they allow the air into respiratory tract, and whenever we are swallowing that one, they close the respiratory tract and the food directly allow into the esophagus. By this way, they regulate the food and swallowing, respiratory and swallowing aspects by this closing or opening by this epiglottis. So this is about that clean picture diagrams which can give some idea about this one. For example, this is an 16th A, means 8 marks question. Simply by mentioning 4 or 5 points we can't get any complete marks. At least try to draw rough diagrams. So no need to mention all this one. Simply here by the trachea, esophagus, here esophagus, we left the trachea. Above this one, how the movement of these, how the posture of these epiglottis, if you show that one, that is enough for get out of 8 marks. So this is about the total question. So how the epiglottis controlled or categorized gaseous and foods by closing their pathways. And now we are enter into another question. What are the secondary metabolites? What are the secondary metabolites? And next, write any alkaloids. Write any 4 alkaloids in a tabular column. So we know that it is very important question. Alkaloids always, each and every question paper we should guess this question. And phyto hormones, and very next to this phyto hormones and alkaloids. These two very, very important. Sometimes either he asks that phyto hormones, or another time he asks that alkaloids. So that's why it is better to thoroughly prepare this answer. For those who are prepared thoroughly on phyto hormones and this alkaloid table, we may guess that one, we have 8 marks in our pocket. It is very important one. And now before that one, he add one more question, that what are secondary metabolisms? So metabolisms are 2 types. They are primary metabolisms and secondary metabolisms. And primary metabolisms, by these primary metabolisms, they produce carbohydrates, proteins and fats. These are the 3 material which are produced by the primary metabolites. By carrying these primary metabolites, carbohydrates, proteins and fats, which are helpful for growth and development, to carry other bodily activity or other metabolic activities. But in these secondary metabolites, so here the major function of the secondary metabolites is for defense mechanism, to protect, to secure itself. They produce some unfavorable taste material from their body. That material can avoid the herbivores away from the plant for their protection. So that's why the type of material which are secreted by the, which are produced by the plants, they call secondary metabolism. The major function of this one, defense mechanism. And what are the some examples for this secondary metabolism? Alkaloids, tannins, resins, gums, latex. So all this mostly they are the nitrogenous compounds. Some of the carbon compounds and some of the nitrogenous compounds are there. These are all the different types of these metabolites, secondary metabolites, which produce defense for themselves. But we find large amount of economical, commercial use of these secondary metabolites which are extracted from the plants. Now if you are continue the question, that is about that secondary metabolism. This flowchart is very, very important to give clarity about this question. And very next, write any 4 alkaloids in a tabular column. So sometimes for example, it is a 4 marks question, we can mention that 4, but it is a 8 marks question. If you already practiced it, we learned many times, we practice many times, we know everything. At the time, better to write all these 8 different types of these secondary metabolites. And if you don't have any time, if you don't have any time, already the time is very near to completed, at the time you don't bother. How many he asked? Only 4. That's why if you are present 4, that is also enough. Now first one we are going to discuss about the common alkaloids which are extracted from the plants. First one, alkaloid in a tabular column, plant, part, use. What are their use? Very first one, quinine, Cinchona officinalis, or it is also called cinchona. And the part what we are using for this medicine, like that one, that is called the common alkaloid, what we are extracted from the bark, and it is used as an anti-malarial drug. Now we discuss about that. Nowadays we are suffered with the COVID problem. So hydroxychloroquine, I think you heard that in news. Hydroxychloroquine, it is a medicine used for malaria. The people those who live in the tropical region, they frequently suffered by the disease called malaria. And all over the world, the people spend much money on the disease of malaria. So hydroxychloroquine, it is one of the medicine for malaria. So here also the quinine, which is used for anti-malarial drug. And second one, nicotine. Nicotine extracted from the plant name called Nicotiana tabacum. And the part which is extracted, that is leaves, it is used as an insecticide. And next, third one, morphine or cocaine, Papaver somniferum, fruit, painkiller. Fourth one, reserpine, Rauwolfia serpentina, it is also called snake plant. We are extracted from roots. It is used for snake bite, medicine for the snake bite. Another one, caffeine, Caffeia arabica, coffee plant. We are collecting that, we are preparing that coffee from the seeds of the Caffeia arabica, and it is stimulate the central nervous system. It stimulates the central nervous system. That's why we heard that every day. If you are make practice to drinking coffee or tea at a particular time, without knowing that one, our body sends some suggestion to better to have some coffee. Why means it is stimulate the central nervous system. And next, another one, nimbin. Nimbin, the scientific name of the plant, which is called Zadiracta indica, neem plant. Nimbin, neem plant. All the total plant of this neem, which are helpful for preparing medicines, even from bark, root, shoot, leaves, fruits, everything from the neem plant, which are helpful to prepare the medicines. So, and all the total plant, a tree, antiseptic, it is used for antiseptic. And another one, scopolamine, or the name of this plant is called Datura stramonium. The alkaloid is called scopolamine. The name of the plant is called Datura stramonium. Fruit and flower, it is act like a sedative. Sedative means they provide some drowsiness. And next one, pyrethroids, chrysanthemum, flower, we are extracted from the flower, it's act like an insecticide. So in this tabular column we find two insecticides. First one is called nicotine, nicotiana tobacco. And next one, pyrethroids, chrysanthemum. These two act like insecticides. By this one, drawing the total, the different types of alkaloids, what he asked, they are the products of the nitrogen compounds. And then if we are maintaining the particular neat label table or neat table, and we write one to another one, it is very good to get good some impression. Those who evaluate your paper by observing this one, by knowing this flow chart, he may impress what we are mentioned, and automatically we get complete marks. So this is the question number 16B, question number 16B, and we can choose either question 16A or 16B, which are easier for us, and depending on the time is also important factor to choose which answer is suitable for us to maintain in a particular proper time. Okay. Now students, we are going to discuss about that section number 4 and 17th question. This last question, it is called diagrammatic question. In this 17A and 17B, he may given two diagrams, one from related to the plants and another from animals. So in this 17th A, in this model paper 4, he asked that one, draw the neat label diagram of human brain. So what we are observing this diagram, this is the human brain, and before we draw a neat label diagram, and at least 4 parts already we mentioned here. So important points, without forget we mention at least 6 and above points. It will be helpful to get good marks. And if you find this one, brain, and I am giving simply explanation about this one, brain is carefully located in a bony case, so that is called cranium, and that brain is covered by the triple membrane layer, that is called dura membrane, arachnoid membrane, pia membrane. Dura membrane, arachnoid and pia membrane. In between the dura membrane, arachnoid membrane, a special fluid flows all over the brain and spinal cord, that is called cerebrospinal fluid. This cerebrospinal fluid, they provide nutrition for the neurons which are present in the brain, and they give protection, and they are helpful to carry their function. And after that one, if you find that brain, a deep groove divide brain into 2 parts, right cerebral hemisphere, left cerebral hemisphere, and then basically brain divide into 3 parts, forebrain, midbrain, hindbrain. All these parts comes under this one. So all the involuntary functions under control of medulla oblongata, and voluntary functions under control of cerebellum. Voluntary functions control of the cerebellum, all the involuntary functions are under control of the medulla oblongata. And this is about the important parts of the brain. So first one, cerebral hemisphere, the division, right cerebral hemisphere, left cerebral hemisphere, 2 cerebral hemisphere we find, and the upper layer which is covered, that is called dura membrane, and middle layer, that is called arachnoid membrane, and final most inner membrane of the brain, that is called pia membrane. In between dura and arachnoid we find the CSF, cerebrospinal fluid, and then brain inside there so many cavities are there. And midbrain, this part is reason shows that midbrain, and the end part of that one, the brain end with the name, that is called spinal cord. So pans varoli, finally they turn into spinal cord. And this is the pituitary gland, master gland, pituitary gland, endocrine gland located here. By drawing the neat label diagram and mention the parts and keep in your box, I hope that you may get out of 88 marks. And now if we are entered into 17B, in this 17B, the diagram related to the plants. So here by observing externally, he said that one, draw the neat label diagram of root hair. Root hair, in the transportation of this one, how the root hair helpful to collect the water and minerals from the external source to the they send into internal one. So here we draw the neat label diagram of these root hair, and we mention here a small flowchart which give idea that how the water and salts, minerals, they collected from the soil to sent into this root hair to inner part of the plant, that is called xylem. Very first the root hair, what I am showing here, this is the root hair, they covered with epidermal tissue, upper layer which is called epidermal tissue, and between the middle layer is called here cortical tissue, and final one the tissue what I am showing this one, endodermis. So epidermis, cortical cells, endodermis cells. By this one 3 crossing that water and minerals, they enter into this root hair, and finally they enter into xylem. By this simple flowchart, we give idea that one how the water and salts, minerals, they transported by with the help of the root hair. And at the same time, and we discuss about that how the root hair collect the water and salts from that outer external sources. Here very first the liquid media which are present in the plant, they called cell sap, and the water content and salts and minerals which are present on external source, they called soil sap. Cell sap means the liquid media which are present inside the plant, and soil sap means the liquid media that present outside the plant. It is an external part. At the time, due to this the process here, lower concentration to higher concentration, the movement of molecule through the semi-permeable membrane. It is an important definition of osmosis. Due to that, osmosis process takes place here. So outside the total, the external sources, that is called soil sap, they have the lower concentration, so they have the higher concentration of water, salts, minerals, whereas inner side they have the low amount of water, salts and minerals. At the time, due to the difference in concentration, osmosis process takes place. By this osmosis process, they carry that through that semi-permeable membrane. Here the root hair membrane, epidermis, cortical cells, endodermis, these three act like a semi-permeable membrane. By crossing these three semi-permeable membrane by the process called osmosis, the water and salts, minerals, they enter into root hair and finally they reach to the xylem. And xylem conduct water, phloem conduct nutrients. By planning this one, by drawing this neat label diagram, we may get 8 marks. So totally we can choose which are easier. For example, here he given the structure of human brain, on another side the root hair experiment. So depending on your practice, depending on your experience, depending on your interest, you can choose any one of that one. You have that complete idea along with that parts. By this one, today we completed the model paper 4. And final question, that is 17th A and B, that is diagrammatic question. Okay, thank you.